Today we will be talking about a very important cell process. It is not only responsible for the world's genetic biodiversity, but also for our gametes, because as we all know, sex sells.
When a sperm and an egg meet, they form a diploid zygote. But what process first created the sperm and the egg? They were formed through a process called
Meiosis. Meiosis 1 and
Meiosis 2.
The process of meiosis is found in many forms of life, such as growing plants, mammals such as the giraffe you see walking here, and aquatic animals such as the goldfish, and even in humans.
But what is Meiosis 1? In Meiosis 1, two diploid cells are created to later be used in Meiosis 2. This is performed by the following steps. It starts with prophase 1. During prophase 1, homologous chromosomes pair and form synapses, a step unique to meiosis. The paired chromosomes are called bivalent, and the formation of chiasmata caused by genetic recombination becomes apparent. Chromosomal condensation allows these to be viewed in the microscope. Paired homologs become physically connected to each other along their lengths by a zipper-like protein structure, the synaptonymal complex. Crossing over, a genetic rearrangement between non-sister chromatids involving the exchange of corresponding segments of DNA molecules begins during pairing and synaptonymal complex formation. Later in prophase 1, microtubules attach to the two kinetochores protein structures at the centromeres of the two homologs. The homologous pairs then move towards the metaphase plate.
Bivalents, each composed of two chromosomes or four chromatids, align at the metaphase plate. The orientation is random with either parental homolog on the side. This means that there's a 50-50 chance for the daughter cells to get either the mother's or father's homolog for each chromosome. Pairs of homologous chromosomes are now arranged at the metaphase plate, with one chromosome in each pair facing each pole. In anaphase, the chiasmata separate. Chromosomes, each with two chromatids, move to separate poles. Each of the daughter cell is now a haploid with 23 chromosomes, but each chromosome has two chromatids. Breakdown of proteins responsible for sister chromatid cohesion along chromatid arms allow the homologs to separate. The homologs then move towards opposite poles, guided by the spindle apparatus. Now, nuclear envelopes may reform or the cell may quickly start meiosis 2.
At the beginning of telophase 1, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis usually occurs simultaneously with telophase 1, forming two haploid daughter cells. In animal cells like these, a cleavage furrow forms. In plant cells, a cell plate forms. No chromosome duplication occurs between meiosis 1 and meiosis 2.
Now for meiosis 2. Meiosis 2 is similar to mitosis. However, there is no S phase. The chromatids of each chromosome are no longer identical because of recombination. Meiosis 2 separates the chromatids, producing two daughter cells each with 23 chromosomes, and each chromosome has only one chromatid. A spindle apparatus forms, but in late prophase 2, chromosomes, each still composed of two chromatids associated at the centromere, move towards the metaphase 2 plate. Due to the crossing over in meiosis 1, the two sister chromatids of each chromosome are no longer genetically identical. The kinetochores of the sister chromatids are attached to microtubules extending from opposite poles, and the spindle fibers attach themselves to the chromosome. The chromosomes are then positioned at the metaphase plate, as in mitosis.
Breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. Nuclei form, the chromosomes begin decondensing, and cytokinesis occurs. The meiotic division of one parent cell produces four daughter cells, each with a haploid set of unduplicated chromosomes. The four daughter cells are genetically distinct from one another and from the parent cell.
These four haploid cells can go on to become four sperms or one ovum with 3 polar products that go into weight.